Hello, and welcome to the podcast lecture on biological safety for the Department of Material Science and Engineering at Johns Hopkins University. The purpose of this video is to acquaint you with basic practices and procedures for working safely with biological materials in material science and engineering labs. This is sort of a new topic in material science and engineering. Not that long ago, most material science and engineering was done on synthetic or inorganic materials. And the advance of biomaterials is new in our department. So for many of us, these practices and procedures that we'll talk about in this lecture are new and somewhat unfamiliar, and therefore deserve, I think, your careful attention. Before we begin talking about biological materials, let me just remind you about a few points of basic philosophy that we hold in our department. Fundamentally, we think that safety is of the highest importance. Said another way, we believe that there is no experiment which is worth jeopardizing your health or your safety or your life or that of someone else. I'm fond of saying it is, after all, only science. A corollary of this is that none of the activities that we do in our laboratories are started without some prudent consideration of their safety aspects. This is not to say that the simplest lab task requires an extensive safety review. It simply says that as we begin our tasks, we think about the safety requirements or the safety aspects of those tasks. If they are tasks that we are familiar with and we've done many times before, the safety review could just be a simple mental checklist of the right procedure and the right personal protective equipment and so on that we need to use to complete the task safely. If the task is something that's completely new to an individual or to a research group or to the department, then perhaps some more extensive consideration of safety aspects should be required. Our goal, after all, is to have no safety incidents that result in any serious injury to anyone in our department or to anyone in our building. The corollary of that is that we would like to have no excuses for any incidents that might actually occur. Said another way, once an accident happens, it's always easy to go back and make an excuse about why that accident happened or why it couldn't have been prevented or perhaps why it wasn't prevented. But we firmly believe that with careful consideration ahead of time, virtually every laboratory accident can be avoided, and that's what we strive for. Before I begin, let me just recommend to your attention this particular book called Biosafety in the Laboratory, published by the National Research Council almost 2 decades ago now. It is available in the Eisenhower Library. Although it is about 20 years old, it has lots of material which is very relevant to working safely in biological laboratories. In this briefing, which is only 20 to 30 minutes long, we can't possibly cover all the important things about working safely with biological materials that you might need to know. So you can, of course, look to your PI and to the members of your lab group for additional training, but you should also seek out outside resources for things that are unclear or that are perhaps beyond the scope of what we'll talk about here. And this book is one that I would particularly recommend. Much of what we will talk about in this briefing is actually drawn from this particular book. Another source of information are the official Johns Hopkins University policies. These are available online from the Health, Safety, and Environment website, and I've just reproduced a portion of one here. These are, of course, official university statements about how things are supposed to be done in our laboratories. And I would recommend them to your attention as well and to that of your principal investigator. If you ever have any questions about health, safety, and environment policies, you can certainly direct those questions to my attention or to the Health, Safety, and Environment Department directly. Let's talk first about biohazards in the laboratory. In a number of our labs now, we are dealing with biological agents of various kinds. It is easy to assume that certain samples, because you know where they came from perhaps or what they are not infectious. But a basic tenet of biological safety is that we assume that all biological materials are potentially infectious or therefore hazardous. The kinds of hazards that we think about are things that are associated with biological cultures, recombinant DNA, and bodily fluids, which may carry infectious organisms along with them. If we think about who's at risk, there's some obvious people who are potentially at risk for infection in a laboratory, and those, of course, include the students and the scientists who work directly in a particular laboratory along with the PI. But we should point out that other people are frequently in our labs as well. So these include custodians and maintenance people who may come into the lab, or security personnel. And then even going beyond that, anyone who passes through the laboratory area of the building is potentially at risk for an infection should it spread from a biological laboratory. So in my opinion, there is an ethical component to dealing with laboratory safety as well, in that if we create hazardous situations, the people who get hurt by those hazardous situations may not necessarily just be us or the person who created the hazardous situation, but could be someone else entirely, and someone who did not sign up for the risk associated with working in a laboratory environment. And we take it upon ourselves as part of our ethical responsibility as conscientious scientists to make sure that we are not putting other people at risk. So let's start out then with some basic guidelines for biological work. First and foremost is the necessity for good personal hygiene, right? Keeping things clean and avoiding transfer of potentially contaminated material, for instance, to your mouth. There are a set of standard biological lab practices that we'll discuss that need to always be followed. You should know the biosafety level of your work, that is the particular risk category that it falls into. How hazardous are the things that you are actually doing or being asked to do? The corollary of that is you should know what the biosafety level of your equipment actually is, and in particular the biosafety cabinet that you work with. What level of protection does it provide? How does it work? What sort of things does it protect against? And what sort of things, very importantly, does it not protect against? And then there will be a set of precautionary measures that we'll talk about, things like personal protective equipment. We always apply these. It is unfortunate if an accident happens and is made worse by the failure to follow standard appropriate precautionary measures. One which I'll mention, which is mentioned in other briefings, was the tragic fire that killed a researcher at the University of California, Los Angeles last year. We could talk about, in the chemical safety part of these lectures, we do talk about how that accident occurred specifically. But one thing that made it worse was the fact that that particular researcher was not wearing a lab coat, a very standard part of protective precautionary equipment. And if she had been wearing a lab coat, then she may very well not have died from that particular accident. Of course, not all failures to use appropriate precautionary measures rise to that same level. But nonetheless, we have these standard procedures, we have standard precautions that we take, and we need to be consistent in how we apply them. In terms of personal hygiene, as I just mentioned, we want to wear lab coats to prevent getting potentially infectious material on us, and other personal protective equipment such as gloves and eyewear as appropriate. We should never have any hand-to-mouth operations in a microbiological lab. This is to avoid the risk of ingesting a potentially infectious organism. Clean nails and clean hair. We, of course, don't want to carry any infectious organisms in that way. No food, drink, or smoking in the lab. These are all excellent ways to transfer organisms from the lab environment into your body. And any broken skin, cuts and scrapes and things like that should be covered, again, to minimize the chance that we transfer hazardous materials into our body. With regard to the importance of good standard laboratory practices, I think it's interesting to note that in roughly 80% of incidents that in which someone in a laboratory environment was infected by an organism, no specific accident or cause of that infection was ever found. That implies that in those cases, the infection happened as a result of simple standard laboratory practices and techniques which were deficient, which is perhaps a little bit alarming until you realize that if you are following good standard microbiological laboratory practices, then you can prevent something like 4/5 of all accidents just in that very simple way. So what constitutes good lab practice? Right? So the first thing is just good habits. Right? We have a set of standard procedures and we always adhere to those standard practices without exception. It's good lab practice to know what the potential hazards are that are associated with your work. We have adopted a set of appropriate biosafety policies for how we do our work. Education, which is part of what we're doing here. Training that it goes on both as part of these video podcasts, but more particularly in the laboratory itself, where you are taught by a member of your research group how to perform the various tasks. And supervision, which can be provided, of course, by the PI. But in terms of generating or creating a safe laboratory culture, it's very important that every member of the lab supervise every other member. And if they notice any sort of deficiency in lab technique or training, they point it out, of course, in a gentle and friendly way, not necessarily in an accusatory way. But this is the way in which we develop an environment in which everyone is expected to adhere to good practices all the time. And that's important for keeping everyone safe. So here are 7 basic rules of standard microbiological practice. First is never pipette by mouth, an excellent way to inadvertently transfer organisms into your body by ingestion. Use mechanical techniques for pipetting. Of course, any potentially infectious material should be handled carefully. Right? We want to avoid spills, and we especially want to avoid the production of aerosols. Aerosols are very small particles of liquid containing infectious organisms. And once you've generated an aerosol, then of course it is very easy to inhale that aerosol and infect yourself by inhalation. So things like forcefully blowing out pipettes to remove excess material are processes that can produce aerosols and should be avoided. Needles and syringes, of course, are sharp and carry the risk of inoculation with an infectious organism. And so, although they are sometimes required, we restrict their use to times when they are absolutely required. They should not be used as a matter of routine unless it is truly essential to the lab procedure to be done. As we've already mentioned, we wear gloves, coats, lab coats, and other personal protective equipment such as eye protection as appropriate for the particular tasks that we're working on. Washing hands is an aspect, of course, of good personal hygiene. We want to wash hands after our laboratory work and after removing gloves. We decontaminate our work surfaces both before and after the work, before to prevent contaminating our work and after to prevent the things that we've done from contaminating something else. And as already mentioned, we don't eat, drink, or smoke in the lab, and we don't store food and drink in biological laboratories. Other good work practices include controlling access to the laboratory by making sure that the laboratory is locked if there's no one there who's working in the lab. We want to dispose of potentially infectious materials properly. We'll talk about this towards the end of this briefing, but in particular, nothing alive should ever go down the sink drain. We decontaminate all of our excess or waste materials before we dispose of them, both solid and liquid. Trash, which contains potentially infectious organisms, should be covered, and we don't go searching through the trash. No sharps on the work surface. This, of course, to prevent the chance of there being an accidental stick. Any accidents or incidents that occur should be reported and should be documented. And it's difficult for me to overstate how important this is. If we have a minor incident, and that incident is reported to the PI and is documented and discussed, then we have an opportunity to learn from that incident and to make sure that similar things don't happen again. If incidents, on the other hand, are covered up, then we lose the opportunity to learn from them, and we run the risk, of course, of then repeating the incident perhaps in a more serious version later on. So it's very important that any incidents, even if they seem minor, should be reported to your PI so that they can be discussed and documented and prevented from happening again in the future. Finally, we also have an insect and rodent control program. This, of course, to prevent the possibility that insects such as cockroaches or rodents would transfer potentially infectious material from a biological laboratory to another place in the building or outside the building. Let's now talk briefly about the various routes of exposure which are listed here. So direct contact is, as its name implies, it's direct contact of potentially infectious material with the skin, which can carry, again, the risk of infection, particularly through cuts or breaks in the skin. The most important thing here is to wear personal protective equipment as appropriate, particularly gloves, lab coats, and potentially eye protection. You can protect work surfaces with disposable absorbent covers, which can then be discarded, disinfected, and then discarded. We follow all procedures, standard microbiological procedures, carefully to minimize the risk of direct contact. As mentioned earlier, we clean and disinfect our work surfaces both before and after work, and we remove gloves after use. In particular, if our gloves are potentially contaminated with any sort of biological material, we want to avoid transferring that biological material to other surfaces where someone else may pick them up. So a good example of this would be wearing gloves that are potentially contaminated and then going to use a computer keyboard. If your gloves are contaminated and use a computer keyboard, and then someone who comes in after you who was maybe not using the lab or working with infectious materials in the lab, but then uses that same computer keyboard, then of course you could transfer the infectious material to that person. And any sort of thing that people might touch, such as doorknobs, telephones, things like that, are all potential opportunities for contamination. So once you're done with your work in the laboratory, remove the gloves, wash your hands, don't wear your gloves around the building and run the risk of spreading that infectious material to other people. The oral route of exposure is ingestion through the mouth. As we've already mentioned, we never pipette by mouth, and there are no hand-to-mouth operations permitted in our lab. Again, no eating or drinking in the lab, and food and drink are stored outside of the lab. Ocular route is exposure through the eye. So if there is a risk of this, then we wear safety glasses or face shields as appropriate. And as already mentioned, we want to avoid the production of aerosols or small droplets, such as by forcefully expelling liquid from pipettes. Inoculation is direct injection. So as we've already mentioned, we only use needles or syringes when there is no feasible alternative to their use. Of course, if we do use them, then we use great care. We work with them very carefully. We keep track of them so that no one gets accidentally stuck. After use, syringes and needles should be disposed of properly in sharps containers, such as the one shown here. I'll point out, I took this photo of in one of the labs in our department, and I was very happy to see the sharps container until Professor Mao pointed out to me that one of the rules with sharps containers is that they are always supposed to be on a metal base. And this particular one is therefore in violation of that particular rule because it doesn't have such a metal base. So both sharps containers and their bases are available from the stores. And so this particular one should be retrofitted with that base, and you should make sure that the sharps container that you're using has such a base. The respiratory route of exposure is breathing in small infectious particles that may be in the air, and primarily this involves the risk of breathing in aerosols. So we want to make sure that if we're doing things like centrifugation or expelling material from pipettes, that we're careful about not to generate aerosols. And in particular, we shouldn't forcefully expel liquid material from pipettes. That can be done more gently. Let's talk briefly about the risk groups associated with biohazards. They're lumped into 4 categories from 1 to 4 in order of increasing risk. So the first risk group are organisms that are not associated with any disease in healthy adults. Risk group number 2 are organisms that are associated with human diseases, but these are diseases now which are rarely serious and for which there are preventative or therapeutic treatments available. And I'll mention that all of the work which is currently being done in the Department of Material Science and Engineering falls into one of these 2 risk groups. The higher risk groups are numbers 3 and 4. Number 3 are organisms that are associated with serious or lethal human disease, and the highest level, level 4, are organisms that are likely to cause serious or lethal human disease. Again, there is currently no work being done in our department in biosafety risk group 3 or 4. What goes along with the risk group are biosafety levels, or BSLs. All biomedical research can be categorized into one of 4 levels according to the agent being handled. The biosafety level then is a combination of the lab practices and techniques which are used in handling those organisms, the safety equipment which is used, and the laboratory facilities. And just like the risk groups, the biosafety levels are in 4 categories in increasing order of level of protection required. The fundamental principle, of course, is to contain the potentially infectious organisms. So that's how we reduce or eliminate our exposure to these organisms, by containing them and using safe techniques for handling them. Containment comes from the design of our facilities, the containment equipment, so things like the pipettes and so on that we talked about, but also good laboratory practices. Right? Our primary barriers are first line of defense when working with potentially infectious organisms, and you can think of this now as a protective envelope that contains the infectious agent, and it's designed to minimize or even eliminate exposure to potentially hazardous biological materials. And the most important item of equipment in this regard is the biohazard safety cabinet, which is shown here. This is in cross-section. There's a sash here. Here is the interior of the cabinet itself. The person working would be standing sort of out here. Air is drawn from the cabinet down through a fan. It's then recirculated up to the top in this particular schematic, where it is then divided into 2 streams. One stream which is recirculated into the cabinet after going through a high-efficiency filter, and the other stream which is vented to the outside, also after going through a high-efficiency filter. It's important to note that biosafety cabinets are different from chemical fume hoods in this regard. In a chemical fume hood, air is drawn out of the fume hood and vented to the outside. and there's essentially no recirculation of air within the fume hood. And the reason, of course, is to remove the hazardous material and prevent exposure of people to that hazardous material. In a biohazard safety cabinet, a significant quantity of the air is recirculated. In the case of our cabinets in our department, it's about 70%. So this means that the biosafety cabinets are not appropriate for working with hazardous volatile chemicals because they stay in the cabinet, and only a relatively small fraction, something like 30% of the air, is actually vented to the outside. The 4 biosafety cabinets in material science and engineering are suitable for work in biosafety levels 1, 2, and 3. But as previously mentioned, all the work currently ongoing in the department is in biosafety levels 1 or 2. Let's talk briefly about how we handled biohazardous waste. Any sort of liquid that contains potentially infectious organisms can be disinfected and disposed of down the drain. As we mentioned earlier, no potentially infectious material should be sent down the drain unless it's first been disinfected. The exception to this is that, of course, organic chemicals, things that are not water-based, cannot go down the drain. They need to be treated as hazardous chemicals. However, so far as I'm aware, none of that sort of work is going on in the department at the present time. Solid hazardous wastes go into containers like this one illustrated down here at the lower left. It's a container. You can sort of see here it has this universal biohazard logo. You can see it's sort of partially obscured by the flap, but you can actually see it over here. Inside the box is a red bag, what we call a biohazard bag. All of the potentially infectious material goes into that red bag. The red bag can be double bagged if you're working carefully, and then sealed inside the box, and the box is then taped up and placed in the hallway where it will be disposed of by the custodians. One thing to note is it's of course always tempting to let the red bag waste box get pretty full and then sort of stuff it closed. That's not a good idea. You should get in the practice of closing these up and sealing them and placing them out for disposal before they are over full. You don't want to be stuffing them so that you run the risk of actually breaking the bag and allowing potentially hazardous materials to be released. In summary, something that I probably should have mentioned earlier is that in the final analysis, you are responsible for your own safety. I can't be there looking over your shoulder all the time. Your PI cannot be there looking over your shoulder all the time. You will be taught safe ways in which to do your laboratory work, but ultimately you are the person who's responsible for carrying out your work in a safe and responsible way. A key aspect of this in the microbiological lab is good personal hygiene and following a standard microbiological practices. Knowing the biosafety level of your work and the level of protection provided by your equipment is also very important. And finally, as I mentioned earlier, the application of appropriate precautionary measures, personal protective equipment such as gloves, lab coats, safety glasses, and so on, is an essential aspect of safety in the department and important for reducing the potential for infection by hazardous organisms in microbiological work. So this concludes the video briefing. Let me just close by reiterating the one point that I mentioned at the very beginning. Safety is very important to us in this department. I think I can speak for all of the faculty when I say that we would much rather have some delay in getting work done as opposed to people rushing ahead to finish work and then having an accident and having someone injured. So if you ever have any questions about the right way or the safe way to do something, or whether or not something you're being asked to do is safe, you should stop and take the time to ask that question of another member of your lab group or of your PI. You can, of course, always come to me and ask questions as well. I'm the department safety officer, and we'll try to handle those questions as expeditiously as we possibly can. But finally, safety is very, very important. Again, there is no experiment which is worth jeopardizing your health or your life or that of someone else. After all, in the final analysis, it is only science. So with that, I thank you for your attention.